Good day to everyone. My name is Darko VONČINA. I work as Associate Professor at University of Zagreb Faculty of Agriculture, Department of Plant Pathology. And on behalf of group of authors, Josip RAŽOV from Syngenta Agro, Silvio ŠIMON from Croatian Agency for Agriculture and Food, I will present you the results of virus screening process done on source cherry Maraska in plantation called VLAČINE. Source cherry variety Marasca is considered as Croatian native variety, and its origins dates to 14th century. It is characterized by excellent fruit quality, strong aroma, and it is mainly used in food and beverage industry for the production of juices, dessert wines, and famous liquor like Maraschino. It is grown mainly in North and Middle Dalmatia, and its gold era dates back to 1975, when the production was estimated to be around 12.5 thousand tons. And today, the estimation of the production goes around 3,000 tons. In addition to Maraska commercial value, data about its viruses dates back to 1980s. So the aim of this survey was to determine the sanitary status of Maraska and infection rates with economically important viruses. And the second aim was to try to select virus-free trees with good agronomic traits for establishing of mother plantations. And in case that we will not be able to find virus-free trees, the plants infected with lowest number of viruses will be selected for sanitation process. Survey was conducted in plantation called VLAČINE, owned by a company called Maraska, and plantation has surface around 200 hectares. Maraska trees are presented with two clones. One is called Brač 6 and the other one Sokolusha. All trees are grown on rootstock Prunus mahaleb, and the collection of sample for laboratory analysis was done in the middle of June, when we selected 200 trees, 50 trees with excellent agronomic traits and very high yield, 100 trees with average agronomic traits and yield, and 50 trees with poor agronomic traits and very low yield. From each tree included in investigation, samples of leaves were collected using collecting scheme north, south, east, west. So here you can see the collecting scheme. So trees were selected from 11 different locations, and the green dots are representing the trees with excellent agronomic traits. The orange dots are representing trees with average agronomic traits and yield, and the red dots are representing trees with very poor agronomic traits and very low yield. So on this slide, you can see our field work, collecting the samples, taking pictures and photos of leaves from each tree, making notes, and also putting the marks on each tree. Viruses included in investigation were those from certification scheme from cherry, defined by European and Mediterranean Plant Protection Organization. So here you can see the list of 10 viruses: apple chlorotic leaf spot virus, apple mosaic virus, rabies mosaic virus, cherry leaf roll virus, petunia asteroid mosaic virus, prune dwarf virus, prunus necrotic ring spot virus, raspberry ring spot virus, strawberry latent ring spot virus, and tomato black ring virus. Additionally to this list, we added plumpox virus, since that virus is very common in Croatia. The presence of all mentioned viruses in cherry trees were checked by ELISA. ELISA negative trees on 11 previously mentioned viruses were additionally tested on 4 viruses: cherry mottle leaf virus, cherry green ring mottle virus, little cherry virus 1 and 2, and for those tests we use reverse transcription polymerase chain reaction. For ELISA, we use commercial kits provided by Loewe Biochemica, and from each tree included in investigation, 0.1 grams of leaf tissue was grinded in mortar using pestle and liquid nitrogen, and all the other ELISA steps were conducted according to manufacturer's instructions. Here you can see the part of ELISA results. In this case, you can see several trees that were positive on Prunus necrotic ring spot virus. In general, trees or samples with OD value at least 3 times the average value of negative controls were considered as positive. And green marks and trees are those that were free of all 11 ELISA tested viruses, and those trees were additionally tested on 4 viruses using reverse transcription polymerase chain reaction. ELISA confirmed the presence of 8 different viruses. Most widespread virus confirmed in 99 trees was Prunus necrotic ring spot virus. Petunia asteroid mosaic virus and prune dwarf virus were confirmed in 10 trees. Roseberry ring spot virus in 7. Cherry leaf roll virus, rabies mosaic virus, and plumpox virus in 3 trees, and apple mosaic virus only in 1 tree. According to RT-PCR results, little cherry virus 2 was confirmed in 6 out of 17 trees. In total, out of 200 tested trees, 11 trees were free of all 15 viruses included in investigation. And in this map, you can see the distribution of different viruses in different parts of plantation. Symptoms observed in the field were necrotic ring spots on leaves, on trees infected with Prunus necrotic ring spot virus, yellowing and reduced growth on trees infected with prune dwarf virus, shoot tips deformations and bark cracking on trees infected with Petunia asteroid mosaic virus, leaf mosaic on trees infected with plumpox virus and apple mosaic virus, and uneven fruit ripening on trees infected with little cherry virus 2. At the end, we can conclude that this survey on sour cherry variety Maraska in plantation VLAČINE confirmed the high infection rate with several viruses, especially with Prunus necrotic ring spot virus, which presence was confirmed in almost 50% of analyzed trees. We confirmed the presence of 9 different viruses out of 15 tested. Luckily, we were able to find 11 trees free of all tested viruses, and those trees were selected as potential mother plants for further pomological investigation and production of virus-free planting material. And also, those trees can serve as a good base for further work on clonal and sanitary selection. Presented results are part of the project improving the quality of sour cherry Maraska planting material by sanitary selection and genetic evaluation, approved and funded by Council for Research in Agriculture, with financial support from Ministry of Agriculture of the Republic of Croatia, Company Marasca, and Nursery Prud. Thank you for your attention, and in case of any questions, feel free to contact me or other authors of this presentation and survey.